Welcome back to microbiology. Today we're going to be covering the internal structures of prokaryotes, particularly bacteria, as well as covering a smidge on archaea and a little bit on how we classify cells as a whole. So with that, we're going to get going first with the contents of the cell cytoplasm. And just a little review, the cytoplasm is the gelatinous solution that's found inside the cytoplasmic membrane. It includes everything inside that membrane. So it would include all of the water and nutrients and enzymes and the DNA, all within the cytoplasmic membrane. On the flip side, we have the cytosol. This would just be the fluid portion. So we're only looking at the liquid, not the major macromolecules within. Overall, its contents is about 70 to 80% water. So as you can see, having water available to the cell is very important for its everyday function. The cytoplasm contains a complex mixture of various nutrients, including sugars, those ringed structures that we talked about in chapter 1, glucose, lactose, a bunch of those carbon, hydrogen, oxygen molecules, amino acids, the building blocks to proteins, as well as salts. Another key internal structure found in bacteria is their chromosomes and plasmids, basically their genetic material. The bacterial chromosome is its hereditary material. So it's the part that's inherited. It would be from the parent cell to its daughter cells. And it is a circular strand of DNA. So typically, when I think of a chromosome, I think of the little X's that we line up with a karyotype for humans. And while it's still the same basis, the structure looks a little bit different. So in bacteria, it's going to be a circle rather than a linear chromosome. Bacteria have no membrane-bound organelles, and along with that, no nucleus. Instead, they will have a nucleoid, which is just a region that is allotted to the genetic material. So there is an area that you would be more likely to find the bacterial chromosome in, so that it's not just strewn throughout the entire cell. Plasmids are also a genetic material you might find in bacteria. They're non-essential DNA though. They're much smaller and they're still in that circular shape. Often we refer to them as being extra chromosomal or kind of an additional chromosome. Plasmids won't contain genes that are essential to the everyday function of the bacteria, very basic things like it wouldn't have the code for molecules involved in metabolism or things like that, but they will provide them kind of, I don't know, superhero-like traits, things like protecting them from antibiotic resistance or the ability to break down certain toxins. Additionally, you would find ribosomes within the cytoplasm of a bacteria, and they are sites for protein synthesis. Ribosomes are comprised of both protein and RNA, and together these two macromolecules form a structure that is used to make protein. So as a little preview to a later chapter, DNA, that double-stranded circular chromosome, is used to make RNA, a single-stranded nucleotide kind of molecule. And then that RNA is used to make protein. So this is the central dogma. And the way that we go from that RNA to protein is with these ribosomes, these macromolecular machines. They're comprised of roughly 60% ribosomal or rRNA and 40% protein. Interestingly, when we think of enzymes or molecules that can catalyze reactions, we typically think of proteins. And I was just reading a study recently that showed if we get rid of the protein component of the ribosome, it will still function, which tells us that the part doing the catalyzing or the enzymatic reaction is, interestingly, the RNA component. The protein is just there to help stabilize the RNA complex. Kind of cool. So ribosomes will be found throughout the cytoplasm. They often will also attach to the cell membrane on the inside. And sometimes you will see chains of ribosomes. So you could see them as kind of lone rangers by themselves, or you could see chains of them all grouped together. And these are known as polysomes. The cell might make a chain of them if they need multiple copies of a protein. So there are some scenarios where maybe you only want one copy of that protein because you're only going to need a little bit. And there might be other scenarios where you need as many as you can make as fast as you can make it. And as we know, assembly lines are much more efficient, and it's kind of the same idea here of having chains of ribosomes all grouped on to the same RNA, chugging along, making a bunch of protein as quickly as possible. Scientists have characterized ribosomes by their subunit density. So if we come over here to the right, you'll notice there's first two subunits that make up a ribosome. We have the large subunit and the small subunit. And you'll see there's kind of three little divots here, another preview to a later chapter. These are going to be different sites that the amino acids will move through. So if we have our large subunit together with the small subunit, then we get our whole ribosome down here. And they're characterized by their subunit density, this 50S, 30S, 70S next to it. The S in each of these is the sedimentation rate. So back in the day, probably 50, 60, 70 years ago, scientists were trying to figure out the differences in our ribosomes. And to do this, they used ultracentrifuges. So they would spin the sample really fast, and based on sedimentation rate, they could determine different molecules. So we have like the large subunit will sediment faster than the small subunit, 50S, 30S. And along with this, you notice it's not like a perfectly mathematical sort of relationship, 50 plus 30 does not equal 70. It's just the sedimentation rate. Okay, so they're not going to add up perfectly, but you do know that both of the small subunits will go together into this large subunit. Some bacteria will also have inclusion bodies and microcompartments. So again, prokaryotes are not going to have membrane-bound organelles, but that doesn't mean that they're not going to need to store nutrients or have some specialized functions. Inclusion bodies can be used to store nutrients to kind of prepare for periods where there may not be as much food availability. So bacteria can kind of stash away extra starches, extra fats, if they're trying to prepare for the dry season or something like that. There are also some bacteria that can hold gases within these inclusion bodies. And this is particularly helpful for like photosynthetic bacteria that you might find in aquatic environments, because if you're doing photosynthesis, you need to be able to see sunshine to make those key sugars and key nutrients. And if you're sinking to the bottom of the lake, it's probably not going to work out too well because light can't get there. So they need buoyancy to be near the surface, and they use gases held in these inclusion bodies to keep them closer to the light. There are also some neat bacteria that can store iron oxide within inclusion bodies. Iron oxide is a magnetic compound that we often use for like the old school compasses, okay? And bacteria use them in the same way. They stash this iron oxide in the inclusion bodies. And since it's magnetic, it can help the bacteria kind of orient themselves towards or away certain regions if they're trying to like migrate to a better area. Bacterial microcompartments, or BMCs, are compartments that have a protein shell around them. So again, they're not membrane bound, but they have proteins on the outside, and it makes basically a little bioreactor where it has protein on the outside, enzymes on the inside, and all the enzymes can work together very efficiently to complete a biochemical pathway, because proximity makes it much more efficient than having all the enzymes randomly strewn throughout the cell. Almost all bacteria are going to have a very strong cell wall. They should have peptidoglycan, whether it's gram-positive or gram-negative in it. And the cell wall is the key feature used to provide bacterial cells with their structure. Now, that said, many bacteria have additional cytoskeletal proteins to further assist in their cell structure. It looks similar to what you would see in eukaryotes, where you might have actin and microtubules and intermediate filaments, but the structure of these proteins is different enough that we can target them with antibiotics. So it might look similar, they might have similar structure, might have a similar function, but their actual sequence is different enough that we can make drugs that will target the prokaryotes and not hurt eukaryotes, which is great when we're treating patients because we want to get rid of the pathogen and not hurt the host or the patient we're trying to treat. These cytoskeletal proteins are often arranged in kind of helical ribbons around the cell, primarily right underneath the cytoplasmic membrane. Some bacteria also form endospores. Endospores are made on an as-needed basis. So bacteria can, if the environment becomes hostile, can form endospores to help facilitate their survival. Endospores, shown here in the picture, is kind of this little like small circular body in the end of the cell, are dormant bodies produced by the bacteria in hostile environments. And hostile environments can vary depending on the cell, but it could be things like there's too much salt, or it's too hot or too cold, or the osmotic pressure is off. Any of those kind of things can make it hostile to their survival. Bacteria can exist in two forms. So we have a vegetative cell, which would be kind of the regular everyday cell that's doing its metabolism and dividing. And then we have the endospore that is kind of the dormant seed that we've saved for later. Sporulation is the process of making endospores. And as you can see in the picture here, the endospore will initially exist inside the vegetative cell. So the vegetative cell will sense that there's a problem. It will form an endospore, and then as the environment continues to deteriorate, it will eventually disintegrate, but the endospore will be left intact. Both gram-negative and gram-positive cells can make endospores, but that said, not all bacteria have this capability. We only see one endospore per bacteria. So you can see here, we only have one, which means this is not a reproductive function. So it's just trying to survive as a species. It's not trying to duplicate or replicate itself, because we're going from one vegetative cell to ultimately one endospore, which means our population hasn't gone up. Two major examples of endospores are botulism and anthrax. So botulism we often see with food production, or like canning, things like preparing tomatoes for later. Endospores can hide in there and can be very toxic when eaten. And anthrax, you may have seen like in old crime movies where they're trying to poison somebody. Anthrax is often the culprit. So here's the steps to forming an endospore. You start out with a vegetative cell that senses something is off, and it begins to deplete its nutrients and kind of hunker down. So it has its cell wall, its cytoplasmic membrane, chromosome, basic, regular, everyday cell. It will, in preparation, duplicate its chromosome and begin kind of separating out its cytoplasm. The cell is separated into two regions. It has a sporangium, which is the vegetative cell component, and a forespore. This is the part that's going to become the endospore. The sporangium will engulf the forespore so that it's fully encapsulated inside of the cell. And it will begin in step 5 here to synthesize layers of extra protection around the endospore. So basically it's like wrapping it in bubble wrap and putting it in a box, trying to hedge its bets to really preserve the endospore and its genetic material. Then it will further continue, and a cortex and an outer layer will be deposited. So you'll see it's kind of like multiple layers have been laid down on it. So we have our purple like cytoplasm with the genetic material, we have a cortex, and we have a cell wall, and we're further laying out all of the components to preserve the endospore for later. Finally, it finishes producing the endospore, and then it can be released. And you can see all the layers it has. It has a core, the cortex, and a nice little coat on it. And it has an exposporangium around the outside. So multiple layers of protection. And then if the conditions improve, it could be a month, a year, 10 years, 20 years in between here. And then when it improves, we'll see germination, in which the endospore will swell because it has water inside of it. It's getting rehydrated again, and it will release the vegetative cell found on the inside, and it can go back to its everyday life. Scarce nutrients, things like not having enough carbon or nitrogen, will often be the triggers for sporulation or entering this cycle. And the sporulating cell is known as the sporangium. So the vegetative cell that's making the spore over here is the sporangium. Endospores will become revitalized when favorable conditions come back. And germination is that reversal. So if we go from not enough carbon and nitrogen, oh, now it's back, then we're going to go through germination and that reversal. This can be triggered by a variety of agents. It depends on the cell, what its exact trigger is, but it could be things like a small organic molecule, a particular amino acid that may be difficult to come by, or a salt. But whenever it senses that more of that is available, it will trigger going back through germination. This germination agent will stimulate the cell to form digestive enzymes, or enzymes that can break down all of those layers to kind of come out of its cocoon. Once the cortex is digested, it will let water flow in, and then the water can rehydrate this core cell, and it will come back to its active state. I have a couple of medically significant bacterial species that form endospores. We have Bacillus anthracis, that is famous for anthrax, which can be used as like a bioterrorist agent, or as I said, in kind of old spy movies. Clostridium tetani, this is tetanus that you may have gotten like a tetanus shot for. It stops lockjaw if you get that shot, but if you are infected with tetanus, it can cause all your muscles to clamp up. This is often associated with things like scraping your foot on a rusty nail, because Clostridium tetani is often found in soil. Clostridium perfringens is a gangrene infection. You might think of like rotting limbs or like very horrible infections with that. Clostridium botulinum is botulism. Again, as I was mentioning, it's affiliated with bad canned food that hasn't been properly sealed. And Clostridium difficile is a gastrointestinal disease that plagues a lot of hospitals. It's shortened. You may have heard of C. diff. It would be Clostridium difficile. Now, it's important to note that ordinary boiling of water or of the sample won't destroy endospores in the same way it would destroy regular vegetative cells, but pressurized steam can be used. So that's part of why when we're preparing canned foods, it will often involve sealing the cans in a pressure cooker or something like that, because we want to get rid of all botulism or anything like that. Okay, so on to our last section here. Archaea are also prokaryotes, but they have some key differences between bacteria that we primarily focused on. So to demonstrate this, there's this lovely table from your book here that compares the 3 domains of life. We have bacteria, archaea, and eukarya. Eukarya, we're going to save for later. We have a whole chapter, chapter 4, devoted to them. We're primarily going to focus on bacteria and archaea for today. Both have circular chromosomes. They can be in bacteria 1, 2, maybe 3. Archaea are only going to have 1. They will have a 70S ribosome, much smaller than the eukarya that you see over here. They will have a unique ribosomal RNA signature sequence here, different across all 3 domains. Archaea is more similar to eukarya in its ability to make proteins, you'll notice, and cell walls are made of peptidoglycan. That is only in bacteria. Archaea will have slightly different cell walls. The cytoplasmic membrane lipids are going to look a little bit chemically different across all 3, and there's going to be no sterols in either archaea or bacterial cell membranes. Archaea and bacteria, since they're prokaryotes, will have no membrane-bound organelles, no nucleus, though they may, across all 3 domains, have a flagella of some kind. Archaea are often extremophiles, so the most likely place that you would bump into them is in the environment in very harsh conditions. So like the picture here on the right, this is a pool at Yellowstone, and part of what gives it the color in the pool is archaea, and this is because they can survive high amounts of sulfur and very hot temperatures. Some key terms to be aware of when we talk about archaea are psychrophilic, which means archaea that love cold temperatures. Okay, so these would be ones that you might find at a polar ice cap. Hyperthermophilic would be ones that love high temperatures. In my example from Yellowstone, would fall into that category. Or halophiles are prokaryotes that love salt. So this could be archaea that you might find in a salt marsh. Microbiologists use Berge's manual for characterizing and classifying new types of bacteria in archaea. There are 4 main categories, and they're primarily categorized based on their cell wall. So we first have our gram-negative bacteria. These would be ones that have a thin layer of peptidoglycan. We have gram-positive bacteria that have a gram-positive cell wall, a very thick layer of peptidoglycan, but only 2 layers on the outside, not 3, like a gram-negative cell. We also have bacteria that lack a cell wall, that are going to just have a much more reinforced cell membrane. And then we have archaea. So with that, that takes us to the end of chapter 3. I'll see you next time.